Hello everyone, welcome to the next video lecture on the topic NGS sequencing analysis. So in this particular lecture we will learn how to install a particular tool called as FastQC and also learn how to analyze a FastQ report. So basically from the previous lecture we have understood some of the basic fundamentals of NGS sequencing analysis, and from the previous video that was uploaded before that we have actually downloaded the FastQC file from the NCBI SRA database, right? So basically it's in the FastQ format. So I will just give you a brief introduction of what is a FastQ format. So basically there are different kinds of formats that are present within a sequence file. It can be an SRA, .SRA format, .FastQ format, or .FastQC format, .SAM or SAM format, or .SBA BAM format. So these are all different kinds of sequencing files that contains the information of each read, the sequence of each read that is generated, and how much qualities bases that are present within a read actually have. So it gives all this information regarding that. So basically in this case what I will do is that I will go to the folder that has earlier installed in our Ubuntu. So basically we know that we have made a new folder, that is an analysis folder. So I will go to the I will open up this folder. You can see that the folder has been installed, and in that I will just search for the list out all the files that are present there. So basically we have FastQ files in there. This is the forward read FastQ file, and this is the reverse read FastQ file. We have discussed what is a forward read and a reverse read in the previous video. If you have, please go through the earlier lecture that we had. So what I want to do is that I will just show you the format of how a FastQ file actually looks like. So I will use a command within the Linux tool. The head command. I will show up the first 10. I will give a 20 lines of this test format. Basically it's a test file, but the format is .FastQ. So and I will type in the host file name in this case. So it will open up this particular file name and it will read the first 20 lines of that test file in this case. So you can see that the first 20 lines in the test files has been opened in this case, not the whole file, because if you are opening the whole file it will take time to read all the file, and you would even if you are going to a word file or test file just to open it normally, what happens is that it will take time and just to load, and it might get stuck in the if your laptop is quite slow and everything. So in this case what happens is that you don't have to open up the test editor. You just open in the Ubuntu terminal, you just open up the first 20 lines of this test file there. You can see that the first 10 reads, I hope it is 10 reads, are actually of length 100 bases. Basically there are length is denoted as 100 bases, and you can see that it starts with like a faster format. Instead of a right facing arrow, it starts with an @ sign followed by the forward read name and the coverage area where it is found and the length of the sequence file there. You can see that afterwards in the after the third line it has the same annotation there, but it is provided by a plus sign, meaning that this denotes the quality of the first file there, quality of each and every basis. So if you are comparing the C, then you have to compare with this. If you are comparing A, then you have to compare with this, and you have to compare each character with the other character in the fourth line. Second line has to be compared with the same character that is present in the fourth line itself, basically. So in case of if you are doing for the sixth one, then it is compared with the eighth one in the same line space and everything. And each alphabet or character actually denotes the quality of the base here. You can see that some is denoted by H, B, F, and F. So you can basically look into Google search and all, actually it denotes the quality score and all based on this character. And if you are seeing like characters like E, F, and these two arrows, it means that it is like non-standard. There is some kind of sequencing error that is appearing in this character. So it might be showing that this quality of this particular base might be low in this case, or it might be some kind of correction there. And if you are looking into some kind of characters like for fix and everything, like it means that there are some adapters that are present here that needs to be trimmed out. So far as we are analyzing the data, we are not seeing any adapters here, which might mean that the adapters were pre-removed from the sequencing data and then the sequencing data was uploaded in SRA. That's why we are not showing much error that this adapter will be present here. So okay, so basically you get a basic idea of how a FastQ format actually looks. So this is the read, this is the quality sequence accordingly that is specified according to the read that is presented there above. Okay, so let's just clear this out, and I will install a analysis tool. Basically it is called as a FastQC. So it actually reads this FastQ file and it provides an output so that it is basically understandable by us, basically what is the overall statistics of this particular FastQ, what we need to do, and overall file that will give. So what I will do is that I will install the FastQC. So in order to install the FastQC, you can either install it using Bioconda, but it is a tedious process. And also if you look into Linux and all, it within the sudo package installer and everything, the FastQC is already inbuilt there. So you can just call it out by using sudo apt install FastQC. So basically it installs it out. You can see that I have already installed it out, so no need to do that. The package is already at the newest version, no need to update it totally. In your case you need to install it out, so please do that. So in order to I will just clear this out also. In order to use a FastQC, what you need to do is that you need to type in the name FastQC, then the folder name. I will type out this both file name within that particular command. So it will just start up the whole process in this case. So it will just take some time. First it will analyze the first folder, then first file, then it will analyze the second reverse read file. Okay, so that is basically complete. So I will just list out what is present here. So you can see that apart from the two FastQ files that were present here for the forward and reverse read, you can see that there is an HTML file and a zip file. So basically what you need to do is that I will just type it in explorer.exe. I will just copy this out and then press enter, and this window will open up. And I will also do for the same one, second read file will open up, and these two pops up in this case. So I will just tell you how to analyze this report. So I have opened it for the forward and read file. So in that case you can identify some of the statistics that is present here. It shows that it is file type is conventional base call file, and it has been encoding is done by Illumina sequencing and all. And total number of sequences that are present, that is the read number that are present here, are this much reads are present here, and total number of bases is this much. The sequence length of the each and every file is about 100. So it is kind of fixed. I am expecting that this file might have been checked from the have been quality check, control check in the initial step itself. That's why like the sequence length is 100 base pair, but some errors are there that needs to be fixed. And also percentage GC content is 66%. So this is quite high in this case, in case of reads and all. But however, if you look into Pseudomonas aeruginosa, which is the model organism that we are doing this whole genome sequencing and all, GC content is quite high in this case. It's natively high in this particular organism, so it's quite expected between 60 to 65%. So some amount of GC percentage we can reduce basically, so that's not an issue. So if you look into for first base pair, you can see that per base sequence quality. If you look into each and every reads, if you are looking into reads of length from 1 to 100, because the 100 is the maximum length there, right? The graph is made accordingly to that. You can see that there is some error, because in some cases it is showing a green tick, and in some cases it is showing a exclamation mark, and other cases it is showing a red bar. So basically all of them which are showing the green tick, basically these are good statistics, like basis in the per sequence quality score is good, per base sequence content is good, per base end content, sequence length, distribution, sequence duplication level, these are all are good. But however, there is an exclamation mark per sequence GC content, which shows a yellow exclamation mark, right? Meaning that it is showing a warning. It's just a warning. It's not saying that the sequence, that particular GC content is too bad in this read file. And also GC content might be due to the presence of adapters, the presence of other genomic contamination that is present within the sequencing when you are doing the sequencing other than the organism that you are analyzing. Or it might be due to a native GC content that is present in the genome of Pseudomonas aerogenus, and it is quite high in case of Pseudomonas aerogenus. So other components we can basically reduce, and let's see how it goes basically. However, in this case you can see that the per base sequence quality and per tile sequence quality is showing a red arrow, meaning that it is a bad. You need to correct it out. So we will be looking into both of that in this case. You can see that in the per sequence case, as the length of the insert size increases, or the read size actually increases, you can see that it actually going into a red area rather than it staying into red area. Up to this area you can see that it's actually good. From that it's going into slightly area, we can say that it's moderate, but when it reaches this area, it reaches into a red area, meaning that there is some kind of issue there that we need to sort out based on the length of the reads that are present in this particular forward read file. And in case of per tile sequence quality, you can see that this is a particular heat map kind of thing, meaning that wherever as the position in reads, if you are considering it as a single read, meaning that at the end of the read you can see that there are several red boxes or rectangles that are present here, or orange or yellow, or some kind of things that are present here. So basically this red color or yellow color, which is a more hot colors, what you call as hot colors, right? And hot colors are cold colors are present there. Cold colors denote the blue, and red hot colors, cold colors denote the blue, and hot colors denote the red. So wherever hot colors are present, it means that the bases that are present at that particular region might be having some problem. You need to remove those bases and all. So basically, if you want to analyze this heat map, I want you to consider this particular entire thing as a particular read, meaning that the first bases start from here 1, and if it is having a 100 base pair, then the 100 base pair is present here, base is present here, first base is present here, and the last base is present here. So if you consider that as a read, meaning that at the ends of the read, especially at this area where yellow color is present, and at this area where red color is present, we need to remove that. Basically we need to trim the bases from the ends. So we have certain tools that could do that basically. So if we are doing that properly, then we could actually clear out or sort out the quality control of this particular report. So that all we need to do in this particular file. Apart from that, all the other bases are actually good basically in this case. In the second file you can see that in the second file you can see almost a similar number of bases are there, 100 base pair, 67% GC content is there. So okay, that we need to sort out. In the same percentage there, as the read length gets increased, the per base sequence quality is also gets increased. This might be due to the porosity of the enzyme or other things like the color presence or overlapping that is present there. However, in case of the per tile sequence quality, you can see that rather than at the end of the insert, the sequence quality is bad at the middle. So we need to basically increase the quality at here by removing some of the sequences there. that also a different kind of tool we have. Basically we have to use two different tools to do that. One is cut adapt, which trims the ends of the insert, and FastQC quality control tool, which actually could reduce the less quality reads or bases that are present in the between the reads and reads and all. So apart from that, all the other things are looking good in this FastQC quantifier. There is no issue with the base sequence content. End content means that if there is a less quality base pair, then it will be denoted by n. Sometimes it cannot read the base at all. So at that position of the insert, instead of a base name adenine, guanine, thymine, cytosine, it will represent as n. So that is okay also. And sequence length distribution, because it's not like the sequence length is varying across a long range. The sequence length is constant up to only 100 bases there. So that's okay. Sequence duplication level, meaning that the sequence length in sequence is actually intact. There is no duplication or copy number changes that are present within sequence, so overrepresent sequence element there. And also adapter content in both cases, as you can see, it's not present. So this gives an idea that this file is already processed, and once processed they are uploaded into a NCB SRI tool. So this could be done as a raw data, and also they haven't done much modification and all, because you can see that there is some problem with the per base sequence quality and per tile sequence quality. But in order to do an SNP analysis, because they have done an SNP analysis, much of the reads need to be preserved. But for the continuation of the training and everything, I think we can go for trimming as well as quality control of the reads and piece and see what we get basically. So yeah, so that's how you read a FastQC file and get to know what to do with it. So that's quite essential in this case, how to analyze a FastQC file and do the necessary stuff so that you get a proper experimental data out of it. Yeah, so I hope you learned a lot from this video, and I will see you in next video. Thank you.